So thank you for joining us today. It's now my pleasure to introduce our speaker, Sharon Bola, Global Product Manager from Bio-Rad Laboratories.
Hello, and thanks a lot for the introduction. Thank you for the opportunity to present today. This is a unique and special topic, and I'm excited to be here and share this with you. I hope you find it interesting. So let's dive into the science that's shaping the future of medicine. I want to begin by discussing cancer. As you're aware, cancer is the leading cause of death globally, and it's very likely that everyone who's tuning in today has been touched by it in some way, directly or indirectly. And various types of cancer necessitate distinct treatment methods, including chemotherapy and radiation therapy. And for treatment, the goal is for both controlled as well as site-specific drug delivery. So for cancer and many other diseases, the question is, how can therapies be targeted to the disease tissues and minimize side effects? So one strategy is SIRT, selective internal radiation therapy, also known as transarterial radioembolization. And this places the therapeutic agent at the site while healthy tissue is spared. So in this example, the radioisotope is a therapeutic agent, and it's being delivered to the liver tumor using a catheter. And the radioisotope needs to be associated with something like a delivery vehicle. And this delivery vehicle will stay put by becoming lodged or embedded in the vasculature. But what is an ideal therapy vehicle? Up top are some examples of carrier materials that have been used for the delivery of therapeutics. And this field is rapidly evolving with several therapeutic agents and carriers that are currently being investigated. Recently, there's been interest in chromatography resins as carriers. And there are some critical factors to consider while selecting the type of carrier. It needs to be biocompatible, meaning that it's doing its job and not causing any adverse reactions. So the carrier needs to be inert and not cause harmful effects once it's inside the body. Particle charge. You need to consider the appropriate charge so that the drug, for example, will be bound or immobilized to the carrier and not leak out. And at the same time, you don't want your therapeutic to be too bound too tightly, because then it won't have any effects. You also need to consider how the resin charge can affect circulation time in the bloodstream. Particle size. The size of carrier can affect its ability to reach and penetrate its target effectively. So in the example of SIRT, microspheres range from 20 to 60 micrometers, and lodge in arterioles, which are less than around 100 micrometers within the tumor. And capillaries surrounding healthy tissues is around 8 to 10 micrometers, so these will not be able to be crossed. You also need to think about the labeling efficiency, or how much therapy per target molecule can be attached successfully and delivered safely while maintaining its effect. And stability of the material is important to prevent failure when it's trapped in the small arterioles. And it also must also stay intact to prevent, for example, a radionuclide from leaching out and getting into non-targeted areas. And finally, material purity is very, very critical. You want highly pure materials, and you don't want to introduce other contaminants that may cause adverse reactions.
Let's talk about chromatography and chromatography resins. Usually when we think about chromatography resins, we think of them as tools to purify biologics for medicines. On the left side is a large manufacturing scale column packed with resin that you typically see in the production of biotherapeutics. Several different resin types exist that we'll cover a little later, but we're going to focus on ion exchange resins and show how they're actually being used as part of the therapy. Here, the cation exchange resin on the right shows a radioactive element attached to the resin, creating a radiation-emitting microsphere that is then delivered to the site of the tumor, and the microspheres, infused through the artery that feeds the tumor, lodge in the tumor's blood vessels, where the radioactive materials release high-energy beta particles that irradiate and kill tumor cells while sparing surrounding healthy tissue. This method delivers radiation locally to the tumor, effectively destroying cancer cells and potentially extending the life or preparing patients for other treatments.
This is the overall chemical structure of the ion exchange resins. So basically you have a polymer made from styrene and divinylbenzene. This is then further treated to bind functional groups. So here on the left are cation exchange resins. For example, you could have sulfonic acid or carboxylic acid groups, and these will typically bind positively charged drugs or radionuclides. And on the right side are anion exchange resins, which have quaternary ammonium groups, and these will typically bind negatively charged groups on the therapeutic. This is what resins look like under a microscope. As you can see, they look like spherical particles, and these are the main resin features. As mentioned, they exist as anion and cation exchangers. They're extensively purified, and they are mechanically and chemically stable against acid, base, and organics. And these can also be autoclaved, and they have, the ones from Bio-Rad have a 5-year shelf life. Moving on to benefits, resin microspheres have a density of 1.6 grams per mil, which is like that of blood and facilitates even distribution in the bloodstream. There's been some imaging studies which show that the depths of penetration by resin microspheres is greater than glass in the hepatic and the hepatic arterial system. And the surface sulfonation makes them non-thrombogenic, meaning that they won't form blood clots. And they have also been shown to have relatively low radionuclide leaching and remains intact. This is a table of our cation exchanger and anion exchange resins that have been used as microspheres. And different cross-linkages as well as particle sizes are available. I'm going to focus today on Aminex 50WX4, and I'll highlight some therapeutic examples of how it's been used. Here we're looking at spheres. Spheres labeled with yttrium-90 are manufactured with Aminex, and these are gaining interest as a treatment option for liver tumors via SIRT. While in most cases hepatic cellular carcinoma cannot be cured, SIRT is a treatment that can extend the patient's survival rate and most importantly can improve these patients' quality of life. It's also been observed that patients with metastatic colorectal cancer who received yttrium-90 labeled spheres had a median overall survival rate of 15 months as compared to 4 to 5 months in the control group. Moving on to other therapies, adenoviral vectors are one of the most promising gene vectors for gene therapy. However, they can cause immune responses, and there's issues with tissue targeting. And there's been several studies that show that adenovirus encapsulated in liposomes can be incorporated with Aminex, and attachment of this complex to an Aminex microsphere can improve gene delivery and reduce therapeutic agent toxicity. When selecting a resin vendor, there are several critical factors to consider. One, the vendor should have a strong quality control system in place and compliance with industry standards like ISO 13485 for medical devices, and GMP is critical. Bio-Rad is ISO 13485, which is much more stringent than ISO 9001. The supplier's market history and successful industry partnerships should also be evaluated. And a vendor should also offer technical support and application expertise to enhance the customer experience. Lastly, to meet supply needs and avoid disruptions, a reliable worldwide distribution network that ensures the security of supply is essential. Bio-Rad's commitment to quality and support ensures the feasibility of using these resins for therapeutic purposes. These are our products that we have developed over the years, and we offer a wide range of beads and chemistries, different formats from bulk resin, 96 well plates, and pre-packed columns with different sizes. Bio-Rad's chromatography resins have been proven effective in numerous case studies, and we've successfully helped our clients with improved product yields, and we've streamlined their bioprocessing workflows. For additional resources, you could read more online, and you can also request printed materials. If you have any questions which we're not able to get to today, or you need more information about the resins, feel free to email us at process@biorad.com, and we'll be more than happy to help you out with samples and
any questions that you might have.
I wanted to thank you for your attention today, and I request that you check out our resources at biorad.com/library, where you can request samples and speak to our scientists and check out our tutorials. We are a worldwide network of experts who can guide you from method development all the way to regulatory filing. And with that, I'd be happy to take any questions that you may have. Thank you.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for Sharon now. So our first question is, what happens to the resin when it's inside the body?
Yeah, I get that question asked a lot. So the resin microspheres are not metabolized or excreted. They actually become a permanent implant in the blood vessels of the liver tumors. And the radioactive isotope that's attached to the resin has a half-life of about 2.5 days. And the entire process is designed to deliver localized internal radiation therapy, which is also known as radioembolization. And this allows for a much higher dose of radiation to the target area than is possible with conventional external radiation.
And our next question is, are there other known treatments that use chromatography resins as a drug carrier?
That's a great question. Yes. For example, in some cough syrups, ion exchange resins are used to bind the active ingredient and then slowly release it into the body. And this helps control the release rate and can also mask the bitter taste of the medicine.
And what quality control measures are in place to make a uniform resin size and density?
That's another great question. Bio-Rad performs rigorous in-process testing to ensure resin uniformity, and we have strict specifications that must be met on the certificate of analysis. For particle size, we do something called HORIBA testing, and this accurately measures the distribution to confirm consistency from lot to lot. And HORIBA particle size testing measures the size distribution of resin microspheres very precisely. It uses a laser technology where a laser beam passes through a resin sample, and the way the light scatters is analyzed, and this allows us to determine the average particle size and the size range and the uniformity of the batch. And this is really important because consistent particle size ensures predictable performance.
All right, well, it looks like we're running short on time. So if we don't get to your questions, we'll be passing them along to Sharon, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll pass those along. The last question for the webcast is, are there efforts to develop next-gen resins with smaller sizes and improved targeting and payload precision?
Right now, we don't, but the Aminex that we offer has been extensively tested and performs reliably and has been used clinically. That said, we're always looking at ways to innovate and support technologies surrounding the resins that we offer.
All right. Well, thank you so much, Sharon, for your time and insights today. It's greatly appreciated. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you will receive a follow-up email providing you with a direct link.